Hi, I'm Walter Isaacson, and this Aspen Ideas Lecture is about the great scientific revolution of our time. I like writing about smart people, but after a while, I learned that smart people were a dime a dozen. What counts is being innovative, to be able to think out of the box, as Steve Jobs said. That's what makes you a revolutionary. Just like Leonardo da Vinci helped bring in the scientific revolution by connecting his experiments and his science and his love for nature with the humanities. And likewise, Benjamin Franklin. Benjamin Franklin helps usher in the Enlightenment in America with his scientific experiments and how he connects it to statecraft. And of course, the great revolution of the early 20th century was the physics revolution, ushered in by Albert Einstein, whose theories in 1905 have its fingerprints on everything from the atom bomb to space travel. And during our lifetime, and in the second half of the 20th century, the great revolution was the digital revolution, ushered in by people like Steve Jobs, who were able to connect the microchip, the computer, and the internet. And when all three of those got connected, you indeed have a digital revolution. And now we're entering into the next great revolution, a life sciences revolution, in which molecules will be the new microchips. We'll be able to encode them and recode them and engineer them to do things like edit our own DNA or help us fight viruses. In order to tell the story of this life sciences revolution, I decided to do it through Jennifer Doudna, a great biochemist at Berkeley. And she worked with dozens of people around the world, but she's a great central character because she helped figure out the structure of RNA, how to use it as a gene editing tool, how to apply it to the coronavirus pandemic, and also how to wrestle with the moral issues. She grew up in Hawaii, and she felt a bit like of an outsider because she was a tall, lanky, blonde girl, and all of the kids in her class in the small town of Hilo, Hawaii, were from Polynesian backgrounds. And she said being an outsider caused her to be more introspective, to try to figure out her place in the cosmos. Just like many of my characters, whether it's Einstein or Steve Jobs, they feel a little bit out of place, and they say, how do I fit in? How do I fit into this cosmos? When Jennifer was in middle school, her father left on her bed a copy of The Double Helix, James Watson's book about the discovery of the structure of DNA. At first she thought it was a mystery tale. You know, she loved mysteries. And she put it aside, and when she read it on a rainy Saturday, she realized she was right. It was a detective story about the hunt for the secrets of life. Jennifer had always been curious, like why does water swirl when it goes around rocks? Something that Leonardo da Vinci was curious about, and even Einstein was. She wondered why the sleeping grass in Hawaii curled up when you touched it. And the double helix explained that there were clues inside of our cells that showed how life worked. That mesmerized her, and she decided that she would become a scientist. Well, her school guidance counselor said, no, girls don't do science. But that caused her to persist. That caused her to become a scientist, to go to Pomona and to study chemistry. She did well in chemistry when she was at Pomona, and she decided she wanted to go to graduate school. She applied to the University of Hawaii. And her father said, well, why don't you apply to Harvard? And she said, well, Dad, I'll never get into Harvard. And he said, you're right. You'll never get in if you don't apply. So indeed, she does apply, and of course, gets into Harvard, where she studies under Jack Szostak. In the 1990s, most biologists were studying DNA. They were part of the Human Genome Project that was going to map all the genes and the sequencing of our DNA. And it was all sorts of the alpha males were involved, people like Francis Collins and Craig Venter and Eric Lander. But Jennifer Doudna and many other women who were studying biology at the time focused instead on RNA. Now, DNA is a more famous molecule. It gets itself on magazine covers, but RNA, it turns out, is more useful. It actually does work. It's the one that goes and gets the code from the nucleus of the cell where the DNA is, and tells our cells how to build proteins. What Jennifer Doudna does is she figures out the structure of certain types of RNA. And by understanding the structure, she finds out how they could replicate themselves, how RNA can copy itself. And that helps answer one of the really big questions in biology, which is, how did life begin on this planet? It begins in the stew of chemicals 4 billion years ago, where some of them jostle together and form the components of RNA, and that RNA begins to replicate itself. So she becomes a specialist in RNA, and when she's in Berkeley, she gets a phone call from another woman biologist named Jillian Banfield, who's trying to figure out why the bacteria that she was studying have these clustered, repeated sequences. Now, these sequences had been discovered by a group of people around the world who were studying bacteria in weird places, like Francisco Mojica when he was a graduate student in Spain studying bacteria in salt ponds, or people in the yogurt companies who were seeing the sequencing of the DNA in the bacterial cultures of yogurts and finding out why do they repeat themselves every now and then. And these clustered, repeated, interspaced sequences became known as CRISPRs. And biologists were trying to figure out what's the purpose of these. Eventually, they discover that these clustered, repeated sequences in bacteria are the way that bacteria take a mugshot of a virus that attacks them. Because if you think we have problems with viruses, man, bacteria have been fighting viruses for 3 billion years. And one way they fight them is they take a little mugshot, a little snippet of the genetic code of a virus, and they put them in these clustered repeated sequences. So if the virus attacks again, boom, they can chop up the virus using a scissors or an enzyme that's guided by a tiny piece of RNA. That's all that CRISPR is. But what Jennifer Doudna discovers is how to repurpose that system in order to edit our own genes, in order to edit human genes. Jennifer Doudna and her partner, Emmanuel Charpentier, a biologist from Paris, met at a conference in Puerto Rico, and they said, let us figure out the exact components of this CRISPR system and see if we can engineer it to target DNA at any spot they want. So what Jennifer Doudna and Emmanuel Charpentier and the graduate students do is they figure out what are the exact components of this system that bacteria have been using to fight viruses, and exactly how can we re-engineer it, just recode the RNA to serve as a guide to cut DNA where we want it to cut. And they do that in June of 2012 and publish what becomes probably the most important scientific paper of the 21st century so far. It has a mouthful of a title, but it shows how you can program RNA guides in order to cut our own DNA. And that's how CRISPR works. After Jennifer Doudna and Emmanuel Charpentier published their paper in June of 2012 showing how CRISPR works, there's a race around the globe of different labs trying to show how you can optimize it to work in human cells. Among the people in that race was a wonderful guy named Feng Zhang, a Chinese-born but raised in Iowa, corn-fed, big smile, works at the Broad Institute of MIT and Harvard. And his former thesis advisor, George Church, the famous geneticist at Harvard, was also in the race. Feng Zhang didn't tell George Church about it, and it caused a rift between them as they found out that they were racing against each other. So in January 2013, all three of the labs, Jennifer Doudna's, George Church's, and Feng Zhang, find how you can use CRISPR and optimize it to work in human cells. George Church and Feng Zhang actually get there 2 weeks earlier, and Feng Zhang applies for a patent and secretly pays to expedite his patent application. So he's given the first patent. This creates a patent war that's still going on between the various labs on who has the right to the patent for using CRISPR. Has become quite a bitter battle. But in the meantime, CRISPR has been used to cure genetic diseases. We've already seen how it can do these miracle things. For example, Victoria Gray, a woman in Mississippi who suffered her whole life from sickle cell. She became the first person on the planet to be cured of sickle cell because they've taken her blood cells, used CRISPR to edit them, and now she's producing healthy blood cells. One of the more interesting people in my book is a guy named David Sanchez. He's a 17-year-old kid who loves playing basketball, except when he doubles over in pain in the middle of the court because he suffers from sickle cell disease. At Stanford, where he's being treated, they've told him, we may be able to use CRISPR not only to cure you, but we can use it in your reproductive cells, your sperm, or early stage embryos of your children, so that we can make inheritable edits that will cure sickle cell, not only in you and your children, but in all of your descendants. We could wipe it out from the human race. And David Sanchez says, wow, that would be great, wouldn't it? And then he pauses and says, well, maybe it should be up to my kids to decide whether they want to have it edited out. And they said, what do you mean? Didn't it cause you a lot of pain? Do you want your kids to suffer the pain? And he says, well, yes, it caused me pain, but it also taught me how to get up off the floor. It taught me persistence. It helped shape my character. And that leads to one of the ethical questions we face. Do we make inheritable edits? And if we do so, how might that change the human species? I found it so amazing that David Sanchez would say these things that I contacted him again later. I said, would you really not want to edit it out of your children? And he says, the more I think about it, the more the answer is yes, I would want to edit it out of my children. And I said, well, what about their character building you talked about? He said, fine, I would try to teach them empathy. I'd try to teach them persistence, but I don't want them to feel the pain. Now, to me, that proves there's no easy answer to some of these ethical questions. We should have first thoughts, like David Sanchez, and second thoughts, and even third thoughts on how we use it. Jennifer Doudna became one of the people trying to figure out our way through these ethical questions, especially through the question of inheritable gene editing. Not long after she helped figure out how CRISPR can be used to edit human DNA, she had a nightmare. And the nightmare is that somebody wanted to learn how to use her technology. And when she walked into the room to meet the person, it was Adolf Hitler. And she recoils and said she woke up and just worried about it for the next few months. And she started gathering scientists from around the world to talk through the legal, the ethical, the regulatory issues, the moral questions we're going to have to face. And they all decided it would be a bad idea right now to use CRISPR in order to make heritable gene edits. It's called a germline edit. It's when you make an edit that will be inherited, and that'll be part of the human species forever. The issue of making inheritable gene edits raises some amazing moral questions that'll be the most important we'll have to wrestle with in the 21st century. If we could edit our children so that they could be taller or stronger, maybe resistant to viruses, have great blood cells so they could be wonderful athletes, or faster twitches in their muscles so they could be sprinters, would we do that? And how would we do things like help memory cells, maybe prevent Alzheimer's, but also increase our own memories? That sounds like it could be very useful, but we have to pause and think. What would happen if the rich could just buy better genes for their children? What if we leave this up to the free market? Then it would not only exacerbate the inequalities we have in our society, it would encode them into the human species. And what about the diversity we have in our species? When I stand on my balcony overlooking Royal Street in the French Quarter, I see people going by who are tall and short and fat and skinny and gay and straight, and trans and different colors and eyes and hair and skin. And that diversity helps make us such a creative species. It also makes us resilient as a species. If we could just allow the free market so everybody could edit what they think was the perfect child, would we edit out some of the diversity in our children? I decided in order to understand CRISPR, I should learn to do it myself. Because one of the things about CRISPR is not all that hard. So I went to Jennifer Doudna's lab, and within 2 days, with the help of a couple of graduate students, I was able to edit human cells. Now, of course, we flushed it down the drain with chlorine, didn't become part of the human species. It's hard to inject them into humans and make it work, but it does show that we're going to have to, as a society, come to a consensus on what we're going to do, because it's going to be hard to regulate these things. After most of the scientific community had come to a consensus that we shouldn't make inheritable edits yet, that we weren't quite ready to do that to the human species, a scientist from China, a young scientist named He Zhuangqi, who had been to some of Jennifer Doudna's seminars and conferences, went back to China and used CRISPR to edit the early stage embryos of what became twin girls. He edited their embryos so that they wouldn't have the receptor for HIV, the virus that causes AIDS. And it not only made them immune to AIDS, or tried to make them immune to AIDS, but it would do so for all of their descendants. Everybody was shocked and pretty upset. The Chinese ended up arresting him. He's now in jail. But after the pandemic, it caused people to think, remind me again, why shouldn't we edit the human species to be less susceptible to viruses? In early 2020, when the pandemic hit, Jennifer Doudna had driven her 17-year-old son, Andy, to the train station to go to a robot building competition in Fresno, California. But she woke up in the middle of the night, nudged her husband and said, we've got to go pick up Andy. I don't want him to be in this conference center, because the pandemic had just started to spread. Berkeley was shutting down its campus. So Jennifer and her husband drove until 5 in the morning, extracted Andy from the robot building competition. He's an only child, so he was like, oh, Mom, what are you doing? But as they left the parking lot, they got a text message saying, robot competition is going to be canceled, all kids should go home. And that's when Jennifer realized that she and the biologists she had been working with needed to turn their attention to the coronavirus pandemic. She gathered scientists at Berkeley and all around the Bay Area. Some of them came to her lab and sat socially distanced in her conference room. Others came in by Zoom, and they discussed, what are we going to do to use CRISPR and our gene editing tools and RNA in order to fight the pandemic? And of course, RNA becomes a great detection technology. It's going to create new ways to find viruses in our system that'll be done instantly with home detection kits that will not only detect coronaviruses, but bacterial infections, maybe the resurgence of cancer. So anything with genetic sequence, there'll be these detection kits that we'll have at our home, and that will bring biology into our lives the way the personal computer brought microchips into our lives. And molecules will become the new microchips. We'll learn how to reprogram them. And that's what we did when we reprogrammed RNA to be a messenger, to tell our cells how to make vaccines against coronavirus. The messenger RNA can be programmed to say, build a tiny fragment of this spike protein that's on a coronavirus. And that's how the RNA vaccines work. It can also be used in the future to directly attack the viruses, to do to the viruses in our system what bacteria do, recognize them and then chop them up. And eventually, we may be able to decide we should do what that Chinese doctor did and make inheritable edits in our species so that we won't have the receptors for certain viruses. In October of 2020, Jennifer Doudna and Emmanuel Charpentier were awarded the Nobel Prize for that 2012 paper that they wrote. I thought it might be too early in 2020 for CRISPR to win the Nobel Prize. After all, another of the prizes that year for physics went to a guy who'd made his discovery 50 years earlier, and he wins the prize. Usually the Nobel Committee takes a long time, but I set my alarm for 4 in the morning just so I could hear the live stream from Sweden. And indeed, when they announced that this year's prize is for about rewriting the code of life, I realized it was going to be given to the CRISPR discoverers. And then I held my breath to say, okay, which of the 3, they usually give it to 3 people, is it going to be? And they said Jennifer Doudna and Emmanuel Charpentier, and that was it. So the 2 of them, for their seminal 2012 paper, end up winning the Nobel Prize. This discovery, as the Nobel Prize Committee said, will bring science into a new epoch. But it's also going to bring humanity into a new era, an era in which we can edit our own genes. That's going to be wonderful. We can cure genetic diseases like sickle cell or cystic fibrosis or Huntington's. Dreadful diseases, we can wipe them out of the human species. And eventually, we'll be able to take on things like Alzheimer's and cancer. And then we're going to have to decide, how far are we going to go with this? Should we use it to design our babies? Should we use it to make inheritable edits to enhance the capabilities of our children? These are questions that can't just be answered by the scientists or by the politicians. I think that all of us have to be part of this discussion. And that's why I wrote this book. And I also wrote this book because, as Jennifer Doudna knows, the mysteries of science can be really beautiful, and at times they turn out to be really useful. Thank you.